Well, good morning, everyone, and thanks so much for the kind invitation to this meeting, to the organizers. Well, let me introduce a little bit a different topic, that is the role of the microbes in the different ecosystems in our health and in nutrition. Well, a poor diet is a key determinant of malnutrition, there is no doubt about, but the problem is much more complex than that. This depends on a number of environmental factors, and of those I would like to highlight our lifestyle and food preference that in principle depends on our daily decisions, but that in practice depend on the economical structure and the overall food systems organization that determine the availability of food, the affordability of food, and the nutritional value of those foods. In addition to that, I would like also to stress the role of biological factors, those factors on which we cannot always work, in contrast to those environmental factors that we can modulate or manipulate in some way, that are our genes, the way these are transcribed and translated into proteins that in the end will define our response to a diet that can vary depending on the population group. And of these factors, I would like to introduce a third one. We have realized that we don't live alone. We are colonized by a large number of bacteria that we call the microbiomes. These are present in different parts of our body, but particularly I would like to talk about those that inhabit the gastrointestinal tract. We have tens of trillions of microbial cells that develop an important number of physiological activities. They provide macronutrients. They also regulate nutrient absorption and the catabolic rate, and for example, the glycemic response to a food. And they also regulate the immune function and the brain development and function. And as a consequence of that, alterations in the composition and function of the microbial communities have been associated to a large number of diseases. And this phenomenon is called dysbiosis. And well, in fact, one of the main roles of our microbes is to process foods and transform these foods in the gastrointestinal tract in a large number of metabolites and chemicals that play important roles in our health. For example, neurotransmitters or short-chain fatty acids that are involved in the regulation of the glucose metabolism. They play also an anti-inflammatory role. They let the colonocyte growth and proliferate and strengthen the gut barrier function, and so on. And in this case, taking into account all the functions that are played by the gut microbiota, we can conclude that the energy and the nutritional value of the food is not determined only by the composition of the food itself, but is also mediated by the interactions between the food we have and the gut microbes we have inside. And finally, they also have an important role in the dietary effects on health. And well, in this context, one of the most important processes is the early colonization of the gut by the gut microbiota. Our gut is initially almost sterile, and it starts being colonized after birth. The bacteria initially comes from the mother and the environment. Later on, these bacteria consume the oxygen and it's replaced by other anaerobic bacteria that can thrive, such as bifidobacteria or Clostridia, that establish in our gut. And later on, when we introduce the complementary food, then a more heterogeneous microbiota is established in our gut, including Bacteroides or Prevotella species. And well, this period of the colonization is considered very relevant because it takes place at the same time as many organs and functions are being developed in the newborn. And these two systems interact and help each other to develop. In fact, this is considered a window of opportunity, this first period of life, in which our microbes can help us to develop different physiological systems. For example, the immune metabolic axis and also the gut-brain axis. Our gut microbes, for example, help us to metabolize the human milk oligosaccharides into energy that is going to feed the developing brain, and the microbes that are in the gut also stimulate the immune system and educate the immune system in such a way it can learn which bacteria are commensal and which bacteria are pathogen, and in this way the immune system start knowing how to respond appropriately to pathogens or commensals. Well, in fact, in recent years, we have understand better what are the characteristics of the proper maturation of the gut microbiota at early life and how disturbances in this colonization process is causally related to malnutrition. And we have learned that by transferring the microbiota of healthy infants into germ-free mice and transferring the microbiota of unhealthy infants with different degrees of malnutrition into germ-free mice. And looking at the phenotypes of these mice, we have realized that the phenotype of the corresponding donor was reproduced, demonstrating that is the microbiota the one that contributes to this impairment in growth rates and metabolic phenotype and bone features in the case of the mice that were harboring the microbiota of malnourished infants. Well, in the context of the My New Gut project, that is a European project we have coordinated in the last years in this area, we have worked in obesity in children, and we have proved how the gut microbiota can act also as a predictor of obesity in childhood. We have followed up a cohort of children, and we took samples at 2 time points. In the follow-up, separated by 4 years, we identify 34 children that remain normal weight and 36 that gain excessive weight gain. And what we realized is that we could classify the children in 4 microbiota clusters according to the coabundance of genera, and that the diversity was reduced in this direction. And this cluster that include the infants that remain with a normal weight were those that have increases in Prevotella, that is a bacterial genus that is specialized in the utilization of fiber, that produce via fermentations or chain fatty acids that seems to be beneficial for metabolic health. And in this cluster, we include most of the infants that remain normal weight. While in the other extreme, in this cluster, where the microbiota show the lowest diversity, we include most of the children that were pre-obese or developed obesity. So it seems to be that this microbiota layout with low diversity predispose the children to develop metabolic complications, because in fact we also observe correlations between this cluster T4 and the increases in pro-inflammatory markers linked to obesity. We also analyzed the dietary patterns. We could identify 5 different dietary patterns, and those that were more unhealthy, those diets rich in carbohydrates or fat, and those diets rich in proteins and high fat, D2 and D5, also co-segregate with the microbiota clusters C3 and C4 that were those that include the children that develop obesity in the time course. So this is saying to us that there is a configuration of the gut microbiota associated with unhealthy dietary patterns that can predict the development of obesity in children. And well, it seems to be that during civilization, during our evolution, and the change in the food production system, we have started losing the healthy microbes, and we have started losing the microbes that confer diversity to our gut microbiota. And we now face the challenge of increasing again the diversity and restoring our gut ecosystem, because this process is linked to the increase in non-communicable diseases. And well, in fact, we can do so by thinking about which dietary components can nourish our healthy microbiota, which agricultural practice can lead us to produce fibers that can be utilized by those healthy microbes. In addition to that, we are starting knowing how to cultivate the intestinal bacteria to produce this bacteria at laboratory level, and we could in the future replenish the gut with the healthy bacteria. In addition to that, well, in this context, in the MineWatt project, we have conducted experiments to demonstrate how we can get that. And for example, we have used specific fibers that are utilized by Bacteroides uniformis, a symbiotic commensal bacteria that is in our gut, and through the administration of both ingredients, we can reverse an obese phenotype and that was induced by a high-fat diet into a lean phenotype, and reduce also the intolerance to glucose. And in fact, this was mediated thanks to the role of these Bacteroides uniformis species in the immune system and its ability to reduce the inflammatory process that is associated to obesity. And well, other authors have also done some studies in this context, and they have proved that specific fibers leading to the production of short-chain fatty acids can reduce the incidence of infections, because the short-chain fatty acids are nutrients of the immune cells that are activated to fight against pathogens. In addition to that, we are discovering also how our gut microbes can produce antimicrobial metabolites that can help us to fight against pathogenic bacteria. And this is an example of a commensal bacteria, Ruminococcus ovium, that was identified in the healthy microbiota of infants and was able to produce a molecule that inhibits the expression of virulent factors by Vibrio cholera. And in addition to that, as I showed you, we can also utilize the information of the microbiome to get a footprint that predict the development of different disorders, so health outcomes. In a way, we can intervene at an early stage prior the development of the disease. And well, in addition to all these studies we have done in relation to the microbiota of humans, in recent years we have been encouraged to do a much more global effort in the microbiome area and start investigating the microbiomes of the different ecosystems that are part of the food production system. These are the marine ecosystem or the plant and soil ecosystems. We are realizing that the microbes that, for example, live in the leaves of the plants, in the case of the coffee plant, for example, can help the plant to resist better the abiotic and biotic stresses. Through the manipulation of these microbes, we can increase productivity, we can increase the safety of the foods produced, and we can also increase the nutritional value. And of these initiatives, I would like to mention a couple of them. One is CIRCLES, where we are involved now, and this will start very soon. And in this initiative, we are going to investigate the associations of the different microbiomes in plants and soil, in the marine ecosystem, and in the humans, and to identify alternatives to modulate those microbiomes in such a way they can perform better in terms of productivity yields, safety, and nutritional quality of the foods. In addition to that, we will investigate how these changes in the production system via manipulation of the microbiomes can impact the quality of the food, but also the human microbiota and its health. In addition to that, we are going to start another initiative that was founded by the European Commission, is the Microbiome Support Project of Horizon 2020, where we have created a huge platform where we are participating not only European scientists, but also scientists from all over the world, India, China, Africa, New Zealand, Canada, and so on. And we will try to define the research agenda in this area, and we will create also a political and scientific platform to enable the dialogue between all parties, to try to translate all these developments into applications that can be used by the society in the field. And well, finally, I would like to summarize some of the key messages of my talk. First, the gut microbiota mediates part of the effects of our diet on health, and we should be conscious that we should also notice not only the human body, but also the gut microbiota inside, because this mediates part of the effects of the diet, and we need to restore the host microsymbiotic relationships that confer these benefits. So this means that we need to redefine what a nutritious food is, in fact, something that is very important in this moment where we have to produce affordable food for everyone. Investigating the relationship between the host and the microbiomes is crucial in the whole food system, because this can be a way to identify microbiome modulation strategies that can help us enormously in the next years to increase the productivity, the affordability, and the nutritious values of foods. Of course, this is an emerging scientific field, and we still need a lot of financial support from governmental and non-governmental institutions. We need to create multi-stakeholder platforms in this area too, if we want to translate the knowledge into applications, and we also need to engage everybody in the creation of knowledge to make this feasible. In addition to that, we need enable regulatory frameworks, and to me, two important futures are the following ones. First, harmonization of the legal paths for the authorization of these new products, and we also need fair intellectual property laws in such a way we can incentivize the innovation, but on the other hand, we can grant the access to the new developments to the society. We also will need in the near future a clear pipeline to assess scientifically how these changes in the food production system and the consumption patterns could affect the human microbiota and human health. And of course, we will have to continue working on educational programs to ensure the acceptability of these new food products, and also continue promoting healthy dietary habits in the population. And that's it. Thank you very much for your attention.
Great.